For 2 billion years, Earth had no oxygen. The sky was orange. The oceans were green. Every breath of air would have killed you instantly. Then something changed. Something small. Something alive. Scientists just found evidence that life was already pumping oxygen into the air 600 million years earlier than anyone thought. And if that terrifies researchers, what they found waiting at the end of the timeline should terrify all of us. We will trace the full story. A poisoned planet, a tiny organism that accidentally broke the world, and a countdown that already started. If you enjoy the ride, subscribe. Let's go. Picture the Earth, 3 billion years ago. Stand on the shore. The water stretches out in front of you, deep and dark green, like a swamp the size of an ocean. The sky above you burns orange, a thick, hazy orange that never clears. The sun pushes through it, dim and red, like a coal buried under ash. There are no birds, no waves slapping against a beach covered in shells, no smell of salt. The smell is sulfur. It fills the air the way smoke fills a room with no windows. You would be dead in under a minute. Every breath you try to take would flood your lungs with methane, carbon dioxide, and nitrogen. The same gases that would choke out a flame would choke out you. Oxygen makes up about 21% of the air you breathe right now. Back then, it made up almost none of it. Scientists confirm this. The early atmosphere held so little oxygen that even a single breath of it would have registered as almost zero on a modern detector. The ground is bare rock, dark, volcanic, cracked. No grass, no moss, no roots pushing into the dirt. The land looks like the surface of a dead planet, but just below the surface of that green ocean, something is alive, tiny, invisible, tough beyond anything you can picture. These are the first life forms on Earth. Scientists call them microbes, which just means microscopic organisms, too small to see. They are single cells, each one smaller than a grain of sand. They have no eyes, no mouths, no way to move toward food the way an animal does. They just sit in the dark, in the heat, pulling energy from the chemicals around them the way a battery pulls electricity from a wire. And the ocean they live in runs hot. Scientists theorize that early ocean temperatures reached around 160 degrees Fahrenheit in some regions. That is hotter than the water that comes out of your kitchen tap at full blast, hotter than a cup of coffee fresh off the stove. These microbes thrived in it. They did not just survive that heat, they needed it. Here is the thing that rewrites everything you probably learned in school. We were taught that life slowly, gradually adapted to Earth, that the planet shaped life. But the evidence scientists have pulled out of ancient rock tells a different story. Life shaped the planet. These invisible organisms, sitting in a boiling green ocean under an orange sky, were about to do something so extreme it would change every ocean, every sky, and every breath taken on Earth for the next 3 billion years. They just did not know it yet. And here is what makes that discovery even stranger. Scientists recently found proof that the change started 600 million years earlier than anyone ever believed. Buried in the soil of South Africa, locked inside rock older than almost anything on the surface of the Earth, a signal was waiting. A chemical fingerprint that should not have been there, a fingerprint left by life. The story of how they found it, what it means, and what it tells us about the fate of our own planet starts with understanding exactly how deadly the early Earth really was, and exactly how much that changed when one tiny organism learned a new trick. The microbes that owned Earth in its earliest years had one rule: stay away from oxygen. To them, oxygen was what carbon monoxide is to you, a silent, invisible killer. The moment an oxygen molecule touched their cells, it would trigger a chain of chemical reactions that tore apart the very structures keeping them alive. Their proteins would break down, their energy systems would collapse. They would die fast and completely. Scientists confirm this. The ancient microbes dominating early Earth were anaerobic, meaning they ran entirely without oxygen. They pulled energy from reactions involving sulfur, iron, and carbon dioxide. Their chemistry was built around a world that had none of the gas you are breathing right now, and they were incredibly successful at it. For over 1 billion years, these organisms ruled every ocean, every shoreline, every patch of warm rock on the planet. They had no competition, they had no predators. The entire Earth was their territory, and they had figured out how to live in conditions that would destroy any modern creature in seconds. Picture a factory that runs entirely on a fuel source that is now considered toxic waste. That is essentially what early life on Earth looked like. The fuel was hydrogen sulfide and dissolved iron. The waste product was sometimes a small amount of carbon. There was no oxygen anywhere in the cycle. Now picture that factory running perfectly, at massive scale, for 1 billion years. These organisms were so adapted to their toxic environment that oxygen itself was the foreign threat. Researchers studying their descendants today, which still exist in deep ocean sediment and volcanic vents, confirm that exposure to even tiny amounts of oxygen can destroy their cellular machinery. They have not changed much in 3 billion years because they have not needed to. But here is where the story gets complicated. Some of those ancient microbes did something strange. Scientists theorize that around 3.1 billion years ago, a group of these organisms began experimenting with a different energy process. Researchers tracked the evolution of 36 protein families tied to oxygen metabolism and found a burst of activity at that exact moment in time. A burst that suggests these microbes were beginning to interact with oxygen in ways their ancestors never had. This is a theory, confirmed by the molecular clock model, and it carries a margin of error across 3 billion years. But the signal is clear enough that most researchers take it seriously. These early experimenter microbes were a tiny minority. They existed in scattered clusters, like isolated neighborhoods in a massive city that everyone else ignores. The rest of the planet's life forms had no idea anything was shifting. They should have paid more attention. Because the microbes building that new chemistry were not just surviving differently, they were building the chemical system that would eventually flood the entire atmosphere with a gas that the dominant life forms on Earth could not survive. The rulers of early Earth were about to be overthrown, and the weapon that would end their reign was being built invisibly in the smallest possible corners of the ocean, one microscopic cell at a time. The place where those early experiments happened is stranger than most people imagine. The ocean floor 3 billion years ago looked nothing like it does today. Drop down past the dark green surface water. Keep going, past the cold layer where sunlight disappears. Down further, into complete blackness, where pressure builds until it would crush a modern submarine. Down to where the ocean floor meets volcanic rock, and the rock cracks open in long splits called hydrothermal vents. These vents pour superheated water into the ocean. Not boiling water exactly, but water that has been heated by magma deep inside the Earth. Loaded with minerals and chemicals and energy, the water that shoots out of these vents reaches temperatures of over 700 degrees Fahrenheit in some places, though it cools rapidly as it mixes with the surrounding ocean. The vents tower above the seafloor like chimneys, and around those chimneys, life clustered. Scientists confirm that hydrothermal vents were active on early Earth and served as one of the primary environments for early microbial life. The chemistry is exact. The minerals these vents release, sulfides, iron compounds, hydrogen, matched perfectly with the energy pathways that anaerobic microbes used. The vents were essentially natural power stations for the first life on Earth. Around each vent, a thick mat of microbes built up over thousands of years, layer on layer. The communities that formed there were structured, organized by chemistry. Different microbes occupied different chemical zones around the vent, the way different stores occupy different floors of a mall. Each zone had its own chemistry, its own energy source, its own waste products. These were the first ecosystems on Earth. They ran in total darkness. No sunlight reached them. No wind moved across them. Sound traveled strangely at that depth. They existed in a world of pure chemistry, a world where energy came from the rock itself and life had adapted to take every scrap of it. Think about that scale for a second. The Mariana Trench, the deepest point in today's ocean, drops about 7 miles below the surface. Early Earth's oceans were not the same depth, but the principle is similar. The first thriving communities of life on this planet existed in a place equivalent to stacking 7 Empire State Buildings on top of each other and then going another mile down, in total darkness, in crushing pressure, in water laced with chemicals toxic to modern life, and they thrived. Some of those vent communities included the microbes that were beginning to experiment with oxygen chemistry. In those hot, dark environments, small amounts of oxygen began to appear in tiny pockets. Scientists theorize these pockets formed through chemical reactions between the vent, water, and the rock, creating trace amounts of oxygen that some microbes learned to use. Those experiments were small, contained. The rest of the ocean had no idea. But the strategy these microbes were developing, the ability to pull energy from sunlight using a process that releases oxygen as a byproduct, was about to move out of the deep and into the shallows. And when it did, the vent communities deep below would become a footnote, because what was coming to the shallow ocean would rewrite the planet on a scale nothing had done before. For a long time, scientists agreed on one thing about Earth's oxygen. It arrived suddenly. About 2.3 billion years ago, cyanobacteria exploded in the shallow oceans, pumped out oxygen, and the atmosphere transformed. Before that moment, essentially no oxygen. After that moment, a planet-wide change. Clean, simple, dramatic. Then a geochemist started digging in South Africa, and the whole timeline cracked. The site was a stretch of ancient rock in a region called the Pongola Supergroup. The rocks there are among the oldest exposed at the surface of the planet. They formed from ancient soils that hardened over billions of years into stone, locking in whatever chemicals were present when they formed. Scientists call these fossilized soils paleosols, and they work like time capsules. Everything that existed in the environment when the soil formed gets trapped inside and preserved. Sean Crow, a geochemist, led the team that analyzed samples from this site. They were looking at rock that formed 3 billion years ago, 600 million years before the Great Oxidation Event was supposed to have happened. What they found inside the rock made the team check their equipment. The chemical signature of oxygen was there. Specifically, they found ratios of chromium isotopes that indicated oxygen had interacted with the soil when it formed. Chromium behaves in a specific way when exposed to oxygen. It shifts between different chemical forms, and those forms leave a permanent signature in the rock. The signature the team found could only have been created by one thing: biological oxygen production. This is confirmed science, published and reviewed. The signal was real. The oxygen concentration was incredibly small. Scientists estimate it was roughly 1/10,000th of what fills the air today, though the exact level is still a theory under debate. But the key point was not the amount. The key point was the source. Chemical reactions alone cannot produce that ratio. The geology of the site, the surrounding rock chemistry, the mineral environment, none of it could generate the oxygen signal without biology. Something alive had been releasing oxygen into that environment 3 billion years ago, 600 million years before the history books said it started. That gap is enormous. 600 million years is longer than the entire span of time that complex animal life has existed on Earth. It is longer than the time between the first fish and today. To push the start of biological oxygen production back by that distance completely reshapes what scientists thought they knew about how life and atmosphere interact. And it raised an immediate question that Crow's team could answer only partially. If life was producing oxygen that early, why did the atmosphere not fill with it? Why did the Great Oxidation Event happen so much later instead of right away? The answer to that question involves something strange. Something that suggests the oxygen was there, alive and active, but deliberately trapped, contained, kept to tiny pockets by forces the planet itself was generating. And the way scientists figured out exactly who was producing that early oxygen required building a clock out of something invisible. Sean Crowe did not set out to rewrite history. He was tracking chemical signatures in ancient rock, the same slow, careful work that geochemists do across years of fieldwork and lab analysis. The Pongala site in South Africa had been studied before. Researchers had looked at these rocks for decades, but the specific chemical analysis Crowe's team applied, focused on chromium isotope ratios, had not been done on samples this old with this level of precision. The field site itself is unremarkable to look at. Flat, dusty, rocky ground, dark bands of ancient stone cutting through reddish soil under a hot sun. The rocks do not glow or hum. They sit there, ordinary looking, the way 3 billion-year-old secrets tend to look from the outside. The team drilled core samples, which are long cylinders of rock pulled from the ground, the way you pull the center out of an apple. These cores preserve the vertical history of the soil, layer by layer, going deeper and older as you move down the cylinder. Each layer is a snapshot of conditions from a different moment in time. When they brought the cores back to the lab and ran the chromium analysis, the signal appeared in layers dated to 3 billion years ago. The chromium isotope ratio was shifted in a direction that only happens when oxygen is present, and the shift was 5 times larger than what any known chemical process without biology can produce. 5 times. That is the number that mattered. 1 or 2 times could be explained by random variation or unusual mineral chemistry. 5 times sits outside any explanation that does not involve living organisms producing oxygen. The team ran the analysis multiple times. They checked for contamination. They compared the results to similar rock formations from different locations. The signal held every time, confirmed. Biology was releasing oxygen into the environment 3 billion years ago. The discovery published and drew immediate attention from the research community. Some scientists accepted it quickly because the method was sound and the result was reproducible. Others pushed back, arguing that the chromium signal could reflect conditions scientists had not fully modeled yet. The debate is ongoing, but the core finding has held up through years of scrutiny. Here is why this matters beyond just a date on a timeline. If life was already processing oxygen 600 million years before the Great Oxidation Event, then the history of life on Earth is far more complex than a simple before and after picture. It means oxygen-producing organisms evolved, spread, and operated for hundreds of millions of years in near total secrecy. Geologically speaking, they left almost no trace except this faint chemical whisper locked in South African stone. And it raises a question that the chromium data alone cannot answer. Who exactly were these early oxygen producers? What were they? Where did they come from? To answer that, scientists could not dig in a field. They had to build something far more unusual. A clock built out of the proteins inside living cells, designed to measure time by counting mutations that happened billions of years ago. The results of that clock pointed to a group of organisms so ancient and so strange that scientists had to invent a new name for them entirely. Numbers in science carry weight when they sit outside the expected range. 1% off from a predicted value is noise. 5% off is worth a second look. 5 times the expected value shuts down every other explanation. When Crowe's team measured the chromium isotope shift in the 3 billion-year-old South African soil, the ratio landed at 5 times the level that chemical reactions alone can produce. That single measurement eliminated every geological explanation the team could construct. Volcanic activity could not do it. Mineral weathering could not do it. Heat and pressure from the rock layers above could not do it. Known ultraviolet light reactions of that era could not do it. The only remaining explanation was biological oxygen production. To understand why that number is so powerful, it helps to understand what chromium does when oxygen touches it. Chromium exists in 2 main chemical forms. One form is stable and stays locked in rock. The other form dissolves in water and moves around, and it does this only when oxygen is present. When these 2 forms separate from each other, they leave a specific ratio behind, like a fingerprint. Scientists can measure that ratio in ancient rock and calculate backwards to determine how much oxygen must have been present to cause the shift. The higher the ratio shift, the more oxygen was involved. A small shift means trace amounts. A very large shift means sustained biological production over time. 5 times the chemical baseline means sustained biological production. The oxygen concentration the team calculated from this signal was tiny by modern standards. Picture today's atmosphere as a swimming pool full of oxygen. The level that existed 3 billion years ago was a single drop in that pool, 1/10,000th of current levels, though this estimate is a theory and the exact number is still being refined. But the size of the amount is secondary to the source. Even a drop, if it came from biology, proves that life had already invented the machinery of oxygen production far earlier than anyone believed. The machinery being a process called oxygenic photosynthesis, the same process that plants use today, the same process that puts oxygen into every breath you take. 3 billion years ago, something was running that process, something alive, in an ocean that should not have had any organisms capable of it yet, according to every model scientists had built before this discovery. And the discovery does not sit alone. It connects to another set of data, built using a completely different method that points to the same moment in time. A method that works by reading the rate at which proteins mutate across billions of years. The way you might measure how fast a clock ticks by counting how many marks accumulate on its face. That method named the organisms responsible. It gave them a date, and it revealed that the first wave of oxygen producers on Earth formed something scientists now call a community of common ancestors, linked by a shared leap in biological chemistry that changed everything that came after. The clock said they existed 3 billion years ago, right where Crow's soil data pointed. 2 independent methods, 2 different types of evidence, same timestamp. A molecular clock does not have hands or numbers, it has mutations. Every time a living organism reproduces, it copies its genetic code, and every time it copies that code, tiny errors slip through. Most of these errors do nothing. A few are harmful and get eliminated. A rare few stick around and get passed to the next generation. These surviving errors, called mutations, accumulate over time at a roughly predictable rate. Scientists figured out that if you count how many mutations separate 2 related organisms, you can calculate how long ago they shared a common ancestor. The more mutations separating them, the further back you have to go to find the branch point. It works like counting the rings inside a tree to figure out its age, except the rings are changes in the genetic code spread across billions of years of reproduction. Researchers Dan Taufik and Patrick Shih use this method to study one specific group of proteins. Proteins tied to oxygen metabolism, the machinery inside cells that either produces oxygen, consumes oxygen, or protects against it. They tracked 36 protein families related to oxygen chemistry across hundreds of different organisms alive today. By mapping the mutation distances between these organisms, they traced the family tree of oxygen metabolism backward through time. The clock stopped at 3 billion years ago. That is when the data showed a clear burst of activity, a sudden expansion in the number of organisms carrying oxygen-related proteins. The burst was too concentrated in time to be gradual drift. Something happened around 3.1 billion years ago that caused a rapid spread of oxygen-related chemistry through the microbial world. This is classified as a confirmed finding based on the molecular clock model, though the exact dates carry a margin of error because protein mutation rates are not perfectly constant over billions of years. The direction of the finding is solid. The precision of the date has a range of uncertainty. What the clock identified at that moment was a group of organisms that Taufik and Shih called the last universal oxygen ancestor. That phrase works like the concept of a last common ancestor in evolution, the organism from which all later members of a group descend. Every living thing on Earth today that uses oxygen in its biology traces back to this early group. Every plant, every animal, every human being. All of it descends from organisms that first developed oxygen chemistry around 3 billion years ago in an ocean that looked nothing like the world we know. The match between Crow's soil data and the molecular clock result was immediate and striking. Two completely different methods, one chemical, one biological, both pointed at the same window in time. That alignment gave both findings far more weight than either would carry alone. But the molecular clock revealed something else, something that explains why that early oxygen did not immediately flood the atmosphere. The organisms were scattered, isolated, surviving in tiny biological islands surrounded by a world that was actively destroying any oxygen they managed to produce. Those islands are stranger than almost anything else in this story. Every living thing that uses oxygen on Earth today shares an ancestor. One group, one moment, one leap in chemistry that changed the direction of life on this planet. Scientists call this group the last universal oxygen ancestor. The name sounds abstract, but the idea is concrete. At some point, roughly 3 billion years ago, a cluster of microbes developed the ability to process oxygen in a meaningful way. They were the common starting point for every oxygen-using organism that ever lived after them. Every plant, every insect, every fish, every mammal, every person, all trace their oxygen chemistry back to those organisms. The proteins inside your cells that help you use the oxygen you breathe carry the same basic structure as the proteins those ancient microbes developed 3 billion years ago. Evolution has modified those proteins extensively over time, but the core machinery is the same. That is confirmed by the molecular clock research. The protein families that handle oxygen in modern organisms all branch back to a common origin point at roughly that 3 billion year mark. Here is a way to picture the scale of that inheritance. Every person who has ever lived on Earth, all the billions of them, used lungs and cells built on chemistry that began in organisms too small to see, living in a hot, dark ocean, surrounded by a world that had never seen oxygen before. The entire tree of animal life grew from that root. The last universal oxygen ancestor was a community of microbes, spread across scattered locations in the early ocean. They were a minority, the dominant life forms of the time. The anaerobic microbes that ran the planet vastly outnumbered them. These oxygen pioneers existed in pockets, isolated environments where conditions allowed their oxygen chemistry to function. Scientists theorize these pockets were places where sunlight reached the shallow water, where the right minerals were present, where the local chemistry supported a different energy pathway. Each pocket was essentially a biological experiment running in isolation. The experiments were fragile. Oxygen is reactive. The moment these microbes produced it, the surrounding chemistry absorbed it. Iron dissolved in the ocean water, grabbed oxygen molecules, and turned into iron oxide, locking the oxygen into solid compounds and pulling it out of circulation. The rocks on the ocean floor soaked up oxygen the way a dry sponge soaks up water. The early Earth was an oxygen sink. It consumed the gas almost as fast as these microbes produced it. That is why the Great Oxidation Event did not happen at 3 billion years ago when the first producers appeared. The planet itself was acting as a massive chemical filter, neutralizing every molecule of oxygen before it could accumulate in the atmosphere. This filter ran for 600 million years. And then something shifted. Something caused the production rate to outpace the destruction rate. Something allowed oxygen to finally start building up faster than the planet could absorb it. The organism responsible for that shift was already in the ocean. It had been waiting, and when conditions finally aligned, it multiplied so fast and produced so much oxygen that no geological filter in the world could stop it. The early oxygen did not spread. Picture a city in a snowstorm. Salt trucks go out immediately, melting the snow as fast as it falls. For hours, the roads stay clear, not because the snow stopped, but because the salt consumed it as quickly as it arrived. The snow was real. It just never got the chance to accumulate. That is exactly what happened to oxygen on early Earth. It was being produced. It was real. But the planet had a chemical salt truck running everywhere at once. The biggest consumer of early oxygen was dissolved iron in the ocean. 3 billion years ago, the oceans held enormous concentrations of iron in dissolved form. Iron reacts aggressively with oxygen. The moment an oxygen molecule appeared in the water, iron grabbed it and formed iron oxide, what we call rust. That rust sank to the seafloor, removing both the iron and the oxygen from circulation at the same time. Scientists confirm this process through the rock record. The seafloor from this period is layered with banded iron formations, thick stripes of rust-colored rock deposited over hundreds of millions of years as this chemical reaction played out. These formations exist on every continent on Earth today, mined for the iron inside them. Every piece of steel in every building and car and bridge around you came from ancient ocean rust, created by the reaction between early oxygen and dissolved iron. Beyond the ocean, exposed rock on land was doing the same thing. Volcanic gases rising from fissures consumed oxygen. Minerals in the rock reacted with it and locked it away. The entire surface of the planet was chemically hostile to oxygen accumulation. Inside all of this, the oxygen-producing microbes survived in oases. Scientists currently debate whether these oases were purely local or part of a globally connected network. The leading view, confirmed through the rock chemistry, is that they were isolated pockets, small regions of shallow water where sunlight reached, where local iron concentrations were low enough that oxygen could briefly accumulate, where the chemistry allowed these organisms to thrive in numbers large enough to sustain a colony. Each pocket was self-contained. The oxygen it produced stayed in that pocket for a while, was used by other organisms in the community, and then was consumed by the surrounding chemistry before it could spread further. The oases operated like tiny sealed bubbles in an ocean that would have dissolved any oxygen that escaped. This situation held for roughly 600 million years. Then, the last of the dissolved iron in the ocean ran out. The volcanic outgassing slowed as the planet cooled slightly. The rock surface that had been fresh and reactive became coated and less able to absorb oxygen quickly. The planet's chemical filter was wearing out, and at exactly that moment, a particular group of microbes in the shallow ocean hit a population explosion. They had evolved a more efficient version of oxygen-producing chemistry. They multiplied aggressively, and they were about to produce oxygen faster than anything on Earth had ever managed before. Cyanobacteria do not look dangerous. Under a microscope, they appear as tiny blue-green threads or dots floating in water, clumping together in thin mats and films. They are simple, single-celled organisms with no brain, no strategy, no awareness of what they were doing. They just ran their chemistry, the same way a car engine just runs its combustion, without knowing anything about the fuel or the exhaust. Their chemistry was oxygenic photosynthesis. Here is the basic version of how it works. The cyanobacterium absorbs sunlight. It takes water molecules from the ocean around it and splits them apart using that solar energy. From the hydrogen it pulls out, it builds sugar molecules it can use for fuel. The oxygen left over from splitting the water gets expelled as waste. One tiny cell. Sunlight in. Sugar kept. Oxygen out. Each individual cell produces almost nothing. A single cyanobacterium releases such a small amount of oxygen that it would take billions of them to make a meaningful impact, which is exactly what happened. Scientists confirm that cyanobacteria multiplied explosively in the shallow oceans roughly 2.45 billion years ago, with some evidence of earlier population surges. The shallow water gave them sunlight. The warmer temperatures and available nutrients fed rapid reproduction. Each generation was faster than the last, and every cell in every generation was producing oxygen around the clock. The ocean surface began to fill, then overflow. For the first time in Earth's history, the rate of oxygen production outpaced the rate of chemical consumption. The iron in the ocean was already depleted by millions of years of rusting reactions. The volcanic gases and mineral surfaces had already absorbed enormous quantities. The planet's filter was exhausted. Oxygen began to enter the atmosphere. The cyanobacteria had no idea they were doing anything unusual. They were doing the same chemistry their ancestors had done for millions of years. But now there was nothing left to stop the result from building up. The atmosphere, which had been a stable methane and nitrogen mix for billions of years, began to change faster than it ever had before. Oxygen is a reactive gas. The moment it entered the atmosphere in quantity, it started reacting with everything it touched, including methane. Methane and oxygen react and destroy each other. Methane burns, essentially, converting to carbon dioxide and water vapor when it meets oxygen. The methane blanket that had kept Earth warm for billions of years began to dissolve. It did not disappear instantly. It took millions of years, but the direction was clear and unstoppable. The planet's greenhouse effect was evaporating, and as the methane thinned, temperatures dropped, not a little. The loss of a global methane blanket is like ripping the insulation out of a house in the middle of winter. The planet had no backup warming system. What came next was so extreme that even the cyanobacteria that caused it nearly did not survive it. Oxygen is a strange thing to call dangerous. You need it to live. Every cell in your body runs on it. Pull it from the air for 3 minutes, and the human brain starts shutting down. Oxygen feels like the most essential, most friendly molecule on Earth. 3 billion years ago, it was the most destructive thing on the planet. The anaerobic microbes that owned Earth at the time had no defense against it. Their proteins were built for a world without oxygen. When oxygen appeared in their environment, it attacked those proteins directly, triggering reactions that shredded the internal structure of the cell. Oxygen did not weaken these organisms. It destroyed them chemically, the same way bleach destroys organic material at the molecular level. To these organisms, the rising oxygen was not a climate shift or an environmental pressure they could adapt to. It was immediate, lethal, and everywhere. But the damage oxygen caused extended far beyond biology. In the ocean, the dissolved iron that had soaked up oxygen for millions of years was already depleted near the surface. As oxygen flooded the deeper layers of the ocean, it hit fresh iron concentrations, and the rusting reaction started again at massive scale. Scientists confirm this through the rock record. Banded iron formations, the thick rust stripes in ancient seafloor rock, were deposited across this entire period. They stretch across every continent, in rock layers spanning millions of square miles. Every stripe is a record of oxygen destroying iron. The rust formed in such quantities that it sank to the seafloor in curtains of heavy particles, building up over millions of years into formations dozens of feet thick. The iron was being removed from the ocean permanently, locked into stone at the bottom of the sea. As this happened, the ocean chemistry changed completely. Iron concentrations dropped, sulfate concentrations rose. The deep ocean, which had been anoxic, completely without oxygen for its entire existence, began receiving oxygen for the first time. The chemical balance of the entire ocean was shifting. On land, oxygen reacted with exposed minerals in the rock. Surfaces that had been a dull grey or brown began staining red and orange as iron oxides formed in the soil and rock faces. The entire planet was rusting. And in the atmosphere, the methane that had blanketed Earth for billions of years was disappearing. Oxygen and methane react and eliminate each other. Every oxygen molecule that rose into the upper atmosphere found methane and converted it to carbon dioxide. The conversion rate accelerated as oxygen concentrations grew. The methane blanket thinned. The warming it provided weakened. Global temperatures began to fall. The planet was losing its heat shield, rusting everywhere at once, and flooding with a gas toxic to most of the life on it. All because a tiny single-celled organism was releasing waste products in the shallow ocean. And the worst was yet to come, because the temperature drop that started with the methane loss did not stabilize at a new, cooler normal. It kept going and going until it reached something that should have been impossible on a planet covered in liquid water. Methane is a powerful warming gas, more powerful than carbon dioxide by a wide margin. A single methane molecule traps roughly 80 times more heat than a single carbon dioxide molecule over a 20-year period. The early Earth was wrapped in a thick methane atmosphere, and that methane was the main reason liquid water existed on the surface at all. The young sun was about 25% dimmer 3 billion years ago than it is today. With a dimmer sun, Earth should have been frozen solid. The only reason it was not was the methane greenhouse effect. That methane blanket compensated for the weaker sun, trapping enough heat to keep the oceans liquid and the surface warm enough for life. When oxygen began flooding the atmosphere in large quantities, it started reacting with the methane. The chemistry is simple. Oxygen and methane meet in the atmosphere, react with the help of ultraviolet light, and convert into carbon dioxide and water vapor. Carbon dioxide is also a greenhouse gas, so the conversion does not immediately crash the temperature. The transition was gradual, but carbon dioxide is vastly weaker at trapping heat than methane. As the methane disappeared and was replaced by carbon dioxide, the warming power of the atmosphere weakened dramatically. The planet was getting the same amount of sunlight from a dim young sun, and it was losing its primary insulation at the same time. Scientists confirm that this process drove the global temperature collapse that followed the Great Oxidation Event. The data from ancient rock formations, preserved chemical signatures, and climate models all align on the same conclusion. The oxygen rise and the temperature collapse happen together, and the chemistry connecting them is straightforward. The planet cooled. At first, ice formed at the poles. Then the ice spread toward lower latitudes. As ice covered more surface area, it reflected more sunlight back into space instead of absorbing it as heat. More reflection meant more cooling. More cooling meant more ice. The cycle fed itself. This kind of self-reinforcing cooling loop is called an ice albedo feedback. Scientists use it to model both ancient and future climate events. Albedo just means reflectivity. Ice has high albedo. Dark ocean water has low albedo. When ice replaces dark water, the planet reflects more energy and absorbs less, driving temperatures lower, regardless of what the greenhouse gases are doing. The loop ran without stopping. Ice advanced from the poles toward the equator, covering land and sea alike. Temperatures dropped to ranges Earth had never experienced during any period in its existence up to that point. The methane that had protected the planet for billions of years was gone. The sun was too dim to compensate. The ice kept coming, and it did not stop at the temperate zones. It did not stabilize at a cold but manageable global average. The feedback loop was too powerful. The ice was too reflective. The sun was too weak. What came next was so extreme that geologists gave it a name that sounds more like a disaster movie than a chapter in Earth's actual history. The planet froze solid. Ice sheets that normally form near the poles and stay there advanced thousands of miles toward the equator. They did not stop at the Arctic or Antarctic circles. They did not stop at the temperate zones. Scientists confirm through rock evidence that glacial ice reached equatorial regions during this period, tropical latitudes, the same zones where today you find jungles and coral reefs and year-round heat, were buried under sheets of ice, potentially thousands of feet thick. Geologists call this event Snowball Earth. The evidence for it sits in the rock record across multiple continents. Ancient glacial deposits, scratched and grooved rocks left by advancing ice sheets, and specific mineral formations that only occur under glacial conditions all show up in rock layers from equatorial regions dated to this period. The signal is global. Every major landmass on Earth carries traces of this event. The average global temperature during the peak of Snowball Earth was likely around -40 degrees Fahrenheit. That is colder than the coldest recorded temperature ever measured in Antarctica today. Across the entire planet, including the tropics, the ocean's surface froze. Scientists debate how deep the ice went on the ocean. Some models suggest the ice reached depths of nearly a mile in some locations. Others suggest open water survived near the equator under a thin ice layer. The full snowball versus slushball debate is still active in the research community, and the exact conditions remain uncertain. What is confirmed is that glacial ice reached the tropics and the planet experienced a climate collapse of extraordinary scale. For the organisms living through it, this was a mass extinction in slow motion. The cyanobacteria that caused the event faced their own crisis. Ice covered the ocean surface, blocking sunlight from reaching the shallow water where they thrived. Without sunlight, photosynthesis stopped. Without photosynthesis, the bacteria that had just transformed the planet began dying back on a massive scale. The anaerobic microbes that survived the oxygen poisoning by retreating to deep water and sediment found conditions slightly more stable. Hydrothermal vents continued operating under the ice, providing heat and chemical energy to the communities that had relied on them for billions of years. The deep ocean was cold and ice-covered above, but active geology below kept some zones survivable. On the surface, life had essentially stopped. The planet sat like this, locked in ice, for millions of years. Models suggest the snowball conditions persisted for somewhere between 10 and 30 million years, though exact durations are still being refined and are classified as estimates based on available data. What ended it was the one geological process the ice could not freeze. Volcanoes kept erupting under and through the ice sheets. Carbon dioxide from volcanic outgassing accumulated in the atmosphere. Carbon dioxide, even weaker than methane as a greenhouse gas, was now the only warming gas available. Over millions of years, it built up to concentrations high enough to begin melting the ice. The melt, when it started, was as violent as the freeze. When the ice melted, the ocean did not return to its ancient green color. It turned red. The end of the snowball Earth period triggered one of the most dramatic chemical events in the history of Earth's oceans. The melting ice dumped enormous quantities of fresh water and dissolved minerals into the sea. The return of sunlight reactivated cyanobacteria populations. Oxygen flooded back into the ocean water at elevated concentrations. All of that oxygen hit something that had been building up during millions of years under the ice: iron. During the frozen period, volcanic activity continued releasing iron compounds into the deep ocean. Without oxygen to react with, the iron accumulated, dissolved, and free in the water, reaching concentrations scientists describe as far higher than anything in the modern ocean. The deep water had become a reservoir of dissolved iron waiting for oxygen to return. When the ice melted and oxygen flooded back in, the reaction started instantaneously across billions of cubic miles of ocean water. Iron and oxygen collided at a scale the planet had not seen before. Rust formed in quantities so large that it physically changed the color of the ocean as it precipitated out of the water. Iron oxide particles, which are the same deep red and brown pigments in rust, fell through the water column in curtains. They sank to the seafloor and accumulated in layer after layer, forming thick bands of red and orange-colored rock. These deposits are called banded iron formations, and they are found on every continent on Earth today. To picture the scale, the largest known banded iron formation in the world is in Australia's Pilbara region, spanning an area roughly the size of the state of Colorado. Geologists mine these formations for the iron inside them. Almost all of the iron ore mined on Earth today comes from these ancient rust deposits. Every piece of steel in every skyscraper, every car, every ship, every bridge on the planet traces back to the moment the ancient ocean turned red. The rusting happened in pulses over millions of years, not in a single dramatic event. Scientists confirm this through the layered structure of the formations themselves. Each band represents a different period of oxygen, flooding, and iron precipitation. The layers alternate between iron-rich rock and silica-rich rock, reflecting cycles of high and low oxygen availability as the atmosphere continued to fluctuate. The ocean was being cleaned. Every time oxygen entered the water, it grabbed iron and removed it permanently. The ocean that had been green for over 2 billion years was chemically changing, one rusting reaction at a time, into something closer to the clear blue water we know today. But the cost of that cleaning was enormous. Every organism that had built its life around iron-rich ocean chemistry was losing its environment. The dominant microbial communities of the ancient world were watching their habitat transform into something unrecognizable. And for the anaerobic microbes that had ruled Earth since its formation, what was coming next was not just a change in water chemistry, it was an extinction. For 2 billion years, the anaerobic microbes had no serious competition. They occupied every available habitat on the planet: deep ocean vents, shallow coastal waters, lake beds, hot springs, exposed rock surfaces at the shoreline. They had evolved dozens of different chemical strategies for extracting energy from their environment. They were diverse, adaptable, and dominant in a way that no organism since has matched. Then oxygen arrived in quantities too large to absorb, and their world ended. The process was not quick by human standards. It played out over millions of years, but in geological terms, it was abrupt. And for the organisms experiencing it, there was no adaptation possible. Oxygen did not apply gradual pressure the way changing temperatures or shifting food supplies do. It chemically attacked the proteins inside their cells. The organisms that encountered elevated oxygen concentrations simply died without the time to develop genetic defenses. This event is sometimes called the Great Oxidation Event, and scientists confirm it as the largest mass extinction in Earth's history, larger than the event that killed the dinosaurs, larger than any of the 5 major mass extinctions that followed. The organisms killed were not diverse animals with complex features. They were single-celled, invisible, and enormously numerous, but their destruction fundamentally altered the living world in a way nothing has matched since. Entire lineages of organisms that had existed for over 1 billion years disappeared. Chemical signatures in ancient rock layers show the sudden absence of molecules that anaerobic microbes produce, replaced by molecules associated with oxygen-tolerant life. The transition appears in the rock like a chemical wall. Before it, one world; after it, another. The organisms that survived did so by retreating. Some moved into environments where oxygen could not reach. Deep ocean sediment below the oxygen penetration depth. Mud at the bottom of lakes and bays. Intestinal environments inside other organisms, which would later become crucial as complex life evolved. Volcanic environments with high temperatures that oxygen chemistry operates poorly in. Others developed biochemical defenses, proteins called antioxidants that can neutralize oxygen molecules before they cause damage. Scientists see this in the genetics of modern anaerobic organisms. Some carry basic antioxidant systems that appear to have developed during or shortly after the Great Oxidation Event, evidence of evolution under intense pressure. The survivors became refugees in their own planet. The organisms that had owned every surface of the Earth for 2 billion years were pushed into pockets and corners, the dark and airless places where oxygen could not follow. And they are still there, under your feet right now, in deep soil layers and rock formations hundreds of feet below the surface. The descendants of Earth's original rulers are alive and operating on the same chemistry their ancestors ran 2 billion years ago. In your digestive system, in deep ocean sediment, in volcanic hot springs, the ancient anaerobic world persists in the margins. The oxygen catastrophe did not eliminate them, it just locked them out of the main stage. But even with the anaerobic microbes pushed back, the oxygen story was nowhere near finished. Because the atmosphere that emerged from the Great Oxidation Event was not stable, scientists recently discovered that the transition to a breathable world took far longer and was far more chaotic than anyone imagined. The anaerobic microbes did not accept defeat quietly. They adapted. The ones that could not tolerate oxygen died in enormous numbers. The ones that survived did so through a combination of location, chemistry, and luck. And in those marginal environments, they built new versions of the world they had lost. Deep ocean sediment became the primary refuge. Below a certain depth in ocean sediment, oxygen from the water above cannot penetrate. The sediment layers compact and consume any oxygen that tries to diffuse downward. Below that boundary, the chemistry reverts to something ancient: no oxygen, sulfur compounds, iron compounds, the same energy sources the earliest life forms on Earth relied on. In these sediment layers, communities of anaerobic microbes thrive today with essentially the same chemistry they ran 3 billion years ago. Scientists who drill core samples from deep ocean sediment regularly find living anaerobic organisms in layers dated to tens of millions of years ago. Some research teams have revived microbes from deep sediment cores that had been metabolically active but completely dormant for millions of years. Simply by giving them the right chemical environment, these organisms were not evolving rapidly during that time. They had no reason to. Their environment had not changed. They sat in the dark, running ancient chemistry, completely cut off from the oxygen world that had taken over above them. Similar refuges existed on land, deep rock formations, where water percolates through cracks in the stone far below the surface, host anaerobic microbial communities that have been isolated for geological timescales. Researchers drilling deep into South African gold mines found living microbes 2 miles underground, in water that had been sealed from the surface for millions of years. These organisms were running chemical cycles that have not changed since before the Great Oxidation Event. Scientists confirmed these findings through both genetic analysis and direct chemical measurement of the surrounding rock and water. The organisms were real, alive, and operating on chemistry that the surface world abandoned billions of years ago. Hot springs and volcanic environments provided another type of refuge. High temperatures naturally limit oxygen chemistry because many oxygen-related reactions slow down significantly in extreme heat. Organisms that lived in thermal vents and volcanic areas experienced less oxygen pressure than those in cooler, shallow water environments. Some of these communities maintained their ancient chemistry while the rest of the planet transformed around them. And then there is the environment closest to you, your own digestive system. The intestinal tract maintains conditions that limit oxygen to the surface layers of tissue. Deep in the gut, anaerobic conditions persist, and the microbial communities that live there are dominated by ancient lineages. Researchers studying gut microbiomes have identified anaerobic bacteria whose closest relatives are organisms from environments dating back billions of years. You carry descendants of Earth's original rulers inside your body every day. The survivors did not just persist, they remained ecologically important. Anaerobic chemistry drives nutrient cycling in ocean sediment, soil decomposition, and gut biology in ways that the oxygen-based world depends on. The ancient world did not disappear, it became the foundation that the new world was built on top of. But the new world, the oxygen world above, was far from stable. Scientists discovered something in the rock record that revealed the atmosphere after the Great Oxidation Event was not the steady, breathable air anyone assumed. It was a mess. The Great Oxidation Event did not deliver a stable, breathable atmosphere. It delivered chaos. Geochemist Chadlin Ostrander and his team wanted to understand exactly how the transition from an anoxic atmosphere to an oxygen-rich one actually unfolded. The standard picture was a relatively clean event. Cyanobacteria multiply, oxygen rises, planet transforms. A step change, before and after. Ostrander's team looked at stable isotope ratios of thallium and sulfur preserved in ancient black shales, a type of fine-grained sedimentary rock that traps chemical signatures from the ocean water it formed in. These isotope ratios change in specific, predictable ways depending on how much oxygen was present when the rock formed. The data from the rock layers, covering the period from 2.45 billion years ago to 2.2 billion years ago, told a story no one expected. The atmosphere was oscillating. Oxygen rose, then crashed back down, rose again, crashed again. The oscillations were not small fluctuations around a stable average. They were large swings, sometimes dropping back to near zero oxygen conditions after periods of significant accumulation. Scientists confirmed this pattern is real and repeated across multiple rock sites on different continents, ruling out local effects. The transition to a permanently oxygenated atmosphere took at least 200 million years of back-and-forth swings before the atmosphere locked into the stable state that eventually made complex life possible. 200 million years. That is roughly the same amount of time that has passed since the first dinosaurs appeared and today. The entire age of dinosaurs, plus all of human history, could fit inside the duration of that atmospheric fluctuation. The mechanism driving the oscillations was a balance, and a fragile one. Cyanobacteria produced oxygen, but on the other side, volcanic outgassing released reducing gases that react with oxygen and remove it from the atmosphere. Methane from anaerobic microbes in the ocean also reacted with oxygen. Exposed fresh mineral surfaces on land absorbed oxygen. The planet had multiple simultaneous oxygen sinks, and they did not exhaust at the same rate. When cyanobacteria had a productive period, oxygen accumulated faster than the sinks could consume it. The atmosphere tipped toward oxygen. Then the oxygen itself killed back some of the anaerobic microbes that were producing methane, which removed one of the competing factors temporarily. Conditions shifted. Volcanic activity spiked in a region. Fresh rock surface appeared from tectonic movement and absorbed oxygen aggressively. The balance tipped back toward anoxic. The atmosphere operated like a seesaw, with uneven weights on both sides, and the fulcrum kept moving. This finding is confirmed through the isotope data, though the exact causes of each individual oscillation are still under investigation, and some remain unknown. What finally tipped the seesaw permanently toward oxygen is still debated. Scientists point to several possible factors, including the exhaustion of reactive minerals, a long-term decline in volcanic reducing gases, and the spread of cyanobacteria into new habitats as the planet cooled. The combination of all three likely worked together over millions of years. The atmosphere that resulted from those 200 million years of chaos was the foundation for everything that came after. Every animal, every forest, every ocean filled with complex life. All of it depended on the atmosphere finally stabilizing, and the evidence for how it stabilized was locked inside something scientists almost never thought to look at. The atmosphere had been flipping between oxygen and no oxygen for 200 million years. Each swing was a gamble. Every time oxygen accumulated, something knocked it back down. Every time it crashed, cyanobacteria slowly rebuilt it. The planet was stuck in a loop, and there was no guarantee the loop would ever break. Think of it like a campfire in the rain. You get the fire going, rain falls, it dies, you light it again, more rain, dies again. After enough cycles, you start wondering if the fire will ever stay lit. For 200 million years, Earth's oxygen fire kept going out. The forces suppressing oxygen were relentless. Volcanic eruptions released sulfur dioxide and hydrogen gas, both of which strip oxygen from the atmosphere chemically. Every major eruption was like a bucket of water on the fire. Freshly exposed rock from tectonic activity presented new mineral surfaces, hungry for oxygen, absorbing it before it could accumulate. Anaerobic microbes in the deep ocean released methane continuously, and methane destroyed oxygen on contact in the upper atmosphere. These were not occasional threats. They were constant. The planet's geology was actively fighting against oxygen stability. Scientists confirmed the scale of this fight through sulfur isotope analysis in ancient rock. Sulfur atoms come in slightly different weights, called isotopes, and their ratios in rock change depending on how much oxygen was present in the atmosphere when the rock formed. In oxygen-rich conditions, sulfur chemistry follows predictable paths. In oxygen-poor conditions, ultraviolet light drives sulfur into unusual chemical forms that leave a distinct isotope signature. The rock record from this 200 million year period shows those unusual signatures appearing and disappearing repeatedly, which is direct chemical evidence of the atmosphere swinging between oxygenated and anoxic states. The data is confirmed. The oscillations were real. Here is the thing that makes this era genuinely terrifying to think about. If the balance had settled differently, The seesaw might have locked in the wrong direction permanently. The geological forces suppressing oxygen were real and sustained. If cyanobacteria had been slightly less productive, or if tectonic activity had delivered more reducing gas to the atmosphere, the oxygen window might have closed. Earth might have remained a methane and nitrogen world indefinitely. No oxygen atmosphere, no complex life, no animals, no forests, no humans. The entire history of complex life on Earth balanced on a 200 million year coin flip, and it almost went the other way. There is evidence in the rock record of periods during this oscillation where oxygen essentially disappeared again for millions of years at a stretch. Extended anoxic episodes that could have become permanent if conditions had reinforced them. They did not. The balance tipped slowly and unevenly toward oxygen. But what finally made it stick is something scientists are still working to fully understand. The clues are buried in ancient ocean water, fossilized inside rock, waiting to be read by the right instruments. What those instruments found in the sulfate chemistry of the ancient sea revealed something about the early ocean that most people have never heard. And it points directly to the moment when the seesaw finally stopped. The battle for Earth's atmosphere was invisible, silent, and lasted longer than most people can imagine. On one side, cyanobacteria, trillions of them in the shallow ocean, running photosynthesis around the clock, releasing oxygen as fast as their chemistry allowed, patient, mindless, relentless. On the other side, the planet's own geology, volcanoes punching through the crust and releasing reducing gases, tectonic plates grinding and exposing fresh mineral surfaces that reacted with oxygen and pulled it out of circulation. The solid Earth, massive and indifferent, absorbing oxygen the way a sponge absorbs water. Neither side was trying to win. There was no strategy, no awareness, no goal. It was pure chemistry, playing out across millions of square miles of ocean and atmosphere at the pace of geological time. Scientists can track this war through the chemical record, and the swings were dramatic. During periods of high volcanic activity, reducing gases flooded the atmosphere fast enough to overwhelm cyanobacterial production. Sulfur dioxide from major eruptions reacted with oxygen directly. Hydrogen from volcanic vents dissolved into ocean water and competed with photosynthesis for chemical resources. The oxygen level crashed. The atmosphere shifted toward anoxic. The unusual sulfur isotope signatures appeared in rock forming during those periods, confirming the oxygen crash was real. During quieter periods of volcanic activity, cyanobacteria gained ground. Oxygen accumulated faster than geology could consume it. Levels rose. Sulfur isotope signatures shifted back toward the oxygen-rich pattern. Rock forming during these windows shows the chemical fingerprint of a more oxygenated atmosphere. Then a new eruption cycle started, and the process reversed. This back and forth left a layered record in ancient rock that scientists compare to tree rings, each layer encoding the atmospheric conditions of the period when it formed. By reading those layers across multiple rock formations on different continents, researchers assembled the picture of a 200 million year chemical war with no clear winner, until the geology started losing. The key shift involved the supply of reactive minerals on Earth's surface. Fresh, unoxidized rock is hungry for oxygen. It reacts aggressively and absorbs huge quantities. But once rock surfaces are fully oxidized, coated in their own reaction products, their capacity to absorb oxygen drops significantly. They become saturated. Over millions of years, the exposed rock surfaces on Earth accumulated oxidation products. Soil chemistry changed as minerals transformed. The capacity of the land surface to absorb oxygen decreased with each passing million years. At the same time, the long-term average rate of volcanic outgassing declined gradually as the planet's interior cooled slightly from its early high temperatures. Fewer eruptions meant less reducing gas. Less reducing gas meant less competition for the oxygen cyanobacteria were producing. The war was not won in a single decisive moment. It was won through exhaustion. The geological side ran out of ammunition faster than the biological side ran out of energy. Cyanobacteria kept producing. The planet's capacity to destroy what they produced kept shrinking. The oxygen accumulated and did not crash back down, and the chemistry of the ancient ocean recorded exactly when that shift became permanent in a signal that scientists found inside sulfate dissolved in rock that was once seawater. Some rocks are not just rocks. They are sealed chemical archives, holding exact records of what the ocean water around them contained millions of years ago. And scientists have learned to read those archives using isotopes, the slightly different weight versions of elements that leave distinct fingerprints depending on the chemistry happening when they formed. Sulfur is one of the most useful of these chemical diaries. Sulfate is a molecule made of sulfur and oxygen that dissolves in seawater. When seawater evaporates, sulfate precipitates out and gets locked into mineral deposits called evaporites. These deposits preserve the sulfur isotope ratios of the seawater at the exact moment they formed. Pull a sample from an ancient evaporite deposit, measure the sulfur isotopes, and you get a direct reading of ocean chemistry from millions or billions of years ago. Scientists have done this across dozens of sites worldwide, building a timeline of ocean sulfate chemistry stretching back over 2 billion years. The results tell the story of how oxygen transformed the ocean from the inside. In the oxygen-poor early ocean, sulfate concentrations were very low. The chemistry that produces sulfate requires oxygen, so without oxygen in the atmosphere and water, sulfate had almost nothing driving its formation. The ancient ocean was sulfate-poor. As oxygen levels rose following the Great Oxidation Event, the oxygen drove oxidation of sulfide minerals in rocks on land. Rain carried those oxidation products into rivers, which delivered sulfate to the ocean. The ocean's sulfate concentration began climbing. By 2 billion years ago, scientists confirmed through the evaporite record that ocean sulfate had reached about 30% of modern levels. That is a massive increase from the near zero starting point, but it is still far below what fills today's ocean. The record shows the sulfate building steadily over hundreds of millions of years as oxygen continued stabilizing. Each step up in the sulfate record corresponds to a period of more stable oxygen. Each plateau or dip reflects a period of oxygen instability or a crash back toward anoxic conditions. The sulfate chronicle in ancient rock follows the same oscillation pattern that the thallium and sulfur isotope data from the previous chapter revealed, but from the ocean chemistry side rather than the atmospheric side. Two completely independent chemical signals, one from atmospheric chemistry and one from ocean chemistry, encoding the same story from different angles. The alignment between these datasets is what gives scientists confidence that the picture of 200 million years of atmospheric instability is accurate. Individual datasets can be misleading. When multiple independent chemical systems record the same pattern, the pattern is real. The sulfate record does something the atmospheric data alone cannot. It shows how the ocean recovered, how the chemical balance of the sea shifted from the ancient iron-rich, sulfate-poor, anoxic deep water system towards something that could eventually support complex life. That transition took time. The ocean chemistry lagged behind the atmospheric chemistry by millions of years in some cases. The deep ocean, far from sunlight and surface oxygen, held on to its ancient chemical character long after the atmosphere had changed above it. But eventually, the oxygen reached the deep ocean too. And when it did, it cleared the way for something the planet had never produced before. Something that required a stable oxygen supply to exist, and that would go on to transform the living world in a second revolution as dramatic as the first. The atmosphere stopped flipping, the seesaw locked. This happened roughly 2.2 billion years ago, though the exact date is still being refined and sits as a confirmed estimate rather than a precise measurement. The oxygen levels stabilized into a range that, while lower than today, was consistent enough to hold. The 200 million year oscillation ended. Something had changed permanently. Scientists are still debating exactly what combination of factors caused the lock. There is no single confirmed answer, and the debate is genuine and ongoing. What researchers have done is identified the most likely contributors, each confirmed in isolation, but their exact combined role is still being worked out. The first factor was the progressive exhaustion of reactive mineral surfaces. Fresh, unoxidized rock had been pulling oxygen out of the atmosphere for billions of years, but by 2 billion years ago, the most reactive surfaces had been transformed. Coated in oxidation products, they lost their capacity to absorb oxygen aggressively. The land surface shifted from an oxygen sink to an oxygen-neutral environment. The second factor was a long-term reduction in volcanic reducing gas output. This is linked to the gradual cooling of Earth's interior. As the planet lost heat slowly over billions of years, volcanic activity, while still intense, produced less overall reducing gas than the highly active early Earth. Fewer reducing gases meant less chemical competition, for the oxygen cyanobacteria were releasing. The third factor, and possibly the most important, was the depletion of iron from the shallow ocean. Iron had been the primary oxygen sink in the ocean for billions of years, consuming oxygen almost as fast as it was produced. By 2 billion years ago, the shallow ocean had been cleared of most of its dissolved iron through the rusting process described earlier. Without iron to consume it, oxygen produced in the shallow water had nowhere to go except up into the atmosphere. These three factors, converging at roughly the same time, created a window where production consistently outpaced consumption. Not dramatically, but enough. Oxygen accumulated past the tipping point, and once it crossed that threshold, the chemical systems that had been fighting it became less effective. A slightly more oxygen-rich environment accelerated the oxidation of remaining reactive minerals. More sulfate formed in the ocean. More oxidizing conditions spread to new environments. The process became self-reinforcing in the direction of stability. Scientists describe this as a transition from a bistable atmospheric system, one that could exist in either an oxygen-rich or oxygen-poor state depending on conditions, to a system that strongly preferred the oxygen-rich state and resisted returning to anoxic conditions. The Earth had committed to oxygen, and what followed was a biological explosion that makes the cyanobacteria revolution look like a rehearsal, because the stable oxygen atmosphere unlocked a type of cellular complexity that had been impossible in the chaotic, unstable chemical environment before. The cells that emerged from this new world were larger, more complex, and capable of things that no life form on Earth had ever managed. They were the ancestors of every complex living thing on this planet, and their arrival changed what life could do permanently. The ocean writes receipts in mineral form. Every time seawater evaporates, it leaves behind the dissolved chemicals that were in it. These chemicals crystallize into minerals that preserve the exact chemistry of that seawater at that moment. Ancient evaporite deposits around the world are essentially receipts from oceans that no longer exist, storing their chemical balances in solid rock for billions of years. When scientists analyze these deposits, they are not guessing at what the ancient ocean contained. They are reading a direct record. The chemistry inside the crystal is the chemistry of the sea that formed it. The sulfate record from 2 billion-year-old evaporites tells the recovery story precisely. Sulfate concentration in ancient seawater at 2 billion years ago sat at about 30% of today's ocean level. That single measurement carries enormous information. Sulfate forms when sulfide minerals in rock react with oxygen and dissolve into water. The higher the sulfate concentration, the more oxygen had been driving that reaction for a sustained period. 30% of modern levels means the ocean had been receiving significant oxygen input for long enough to transform a substantial portion of its sulfide chemistry. That transition does not happen quickly. Building sulfate to 30% of modern concentration required millions of years of consistent oxygen availability. The measurement confirms that by 2 billion years ago, the Great Oxidation Event had been real, sustained, and chemically thorough. The ocean was not just experiencing trace oxygen, it was being systematically transformed. But 30% also means the transformation was nowhere near complete. Today's ocean carries roughly 3 times that sulfate concentration. The gap between 2 billion years ago and today represents another 2 billion years of continued oxygen processing, slow and steady, continuing right up to the present. The recovery was not a single event. It was a multi-billion-year project that is technically still running. Scientists also use the sulfate record to cross-check oxygen level estimates from other methods. When the isotope data from atmospheric rock, the thallium measurements from ocean sediment, and the sulfate concentrations from evaporites all align on the same timeline, the confidence in that timeline grows substantially. Each dataset is an independent measurement of the same historical reality. The 3 signals align. 2 billion years ago, oxygen had stabilized enough to begin transforming ocean chemistry in a measurable and sustained way. The oscillations were over. The seesaw had locked. And inside that newly stable chemical environment, something was happening to the structure of cells themselves. Life was not just surviving in the oxygen atmosphere. Life was being physically rebuilt by it. The oxygen that had been a poison to the ancient world was now a construction material. Cells were using it to power more complex internal chemistry than had ever been possible before. The size of individual cells was growing. Internal structures were developing. The raw biological material for multicellular life was being assembled, one chemical reaction at a time. What those new cells became and how long it took them to organize into the first complex organisms is a story that depends entirely on the oxygen stabilization that the ancient seawater receipts confirm happened 2 billion years ago. Without that stabilization, none of what came next was possible. Every animal that has ever existed on Earth owes its life to bacterial waste. That is not a dramatic way of saying something ordinary. It is the literal chemical history of complex life. The oxygen that powers every animal cell on the planet was released as a metabolic byproduct by cyanobacteria that had no idea they were building anyone else's future. Before the Great Oxidation Event and the atmospheric stabilization that followed, complex cells could not exist. The chemistry required to build a large, organized, multi-part cell requires stable oxygen. Without it, the energy available to a single cell is drastically limited. Anaerobic chemistry is efficient enough to sustain tiny, simple organisms, but it cannot power the kind of cellular machinery that leads to eyes, muscles, brains, or anything else found in complex animals. Scientists confirm this through both biochemistry and the fossil record. The first evidence of large, complex cells in the fossil record appears after the Great Oxidation Event, timed closely to the period when atmospheric oxygen stabilized into a consistent range. The correlation is direct and confirmed across multiple independent lines of evidence. The reason comes down to energy density. Oxygen-based respiration produces roughly 18 times more energy per unit of food than anaerobic metabolism. That energy surplus is what allows cells to grow larger, maintain more complex internal structures, develop specialized functions, and eventually organize into multicellular organisms. 18 times more energy is the difference between a flashlight and a floodlight. Anaerobic life built everything it could with a flashlight. Oxygen handed the biological world a floodlight, and complex life was what happened next. The progression from stable oxygen to complex cells to multicellular organisms took hundreds of millions of years. It was not fast even by geological standards, but the direction was set the moment oxygen stabilized. The energy was available. Life used it. Every branch of the animal kingdom, every plant, every fungus, every complex life form on Earth descends from organisms that made use of the oxygen cyanobacteria released. The evolutionary tree of complex life is built on a foundation of bacterial pollution. Humans are, in the most direct chemical sense, made possible by microbes that lived 2.5 billion years ago, producing waste that no one wanted, transforming a planet they could not perceive, enabling a future they could not imagine. And the evidence for all of this is preserved not just in the living chemistry of every cell on Earth, but physically inside ancient rock, in a form so unexpected that scientists almost did not think to look for it. Some of the most precise chemical records from Earth's ancient past are not stored in obvious places. They are stored in salt. When ancient seas evaporated in shallow basins along the edges of early continents, they left behind thick deposits of mineral salt. These deposits formed over thousands of years as water levels dropped, crystallizing out all the dissolved chemicals in the seawater. The crystals grew slowly, trapping tiny pockets of the actual seawater inside them as they formed. Those trapped pockets are called fluid inclusions. They are microscopic bubbles of the original seawater, sealed inside salt crystals and preserved for hundreds of millions or billions of years. Scientists crack open those crystals today and analyze the fluid inside. They are reading ancient ocean water directly. The chemistry inside fluid inclusions from 2 and 3 billion-year-old rock salt deposits tells researchers the exact concentrations of dissolved gases, minerals, and chemical compounds present in those ancient seas. Oxygen levels, sulfate concentrations, dissolved carbon dioxide, all of it preserved in the fluid inside the crystal. This method confirmed the sulfate buildup story from the previous chapter with a level of directness that isotope ratio analysis alone cannot match. Isotope ratios require interpretation and modeling. Fluid inclusions from salt crystals are the actual water, analyzed directly. Scientists confirmed the oxygen signal in ancient salt samples matches the timeline built from other methods. The two datasets align. The story is real. But the fluid inclusions carry additional information that other methods cannot capture. They preserve the organic chemistry of the ancient seawater, the dissolved biological molecules produced by microbes living in those ancient seas. Scientists can identify specific organic compounds in the fluid that act as biosignatures, chemical fingerprints of specific types of organisms. These biosignatures inside ancient salt crystals have confirmed the presence of cyanobacteria in seawater predating the Great Oxidation Event. They have confirmed the presence of other microbial groups at specific time periods. They are a biological census of the ancient ocean, sealed in salt and waiting for the right instruments to read it. The precision of this method continues to improve as analytical technology advances. Researchers today can identify individual organic molecules from samples billions of years old. A decade ago, that resolution was impossible. In another decade, the detail available from fluid inclusions may be fine enough to reconstruct the metabolic chemistry of individual microbial communities in the ancient ocean. The ancient world is not gone. It is archived, written in salt crystals scattered across every continent on Earth, waiting to be read. And that archive does not just tell us where we came from, it tells us what we are heading toward. Because the same chemical logic that transformed Earth from anoxic to oxygen-rich is now running in reverse, driven by a force that cyanobacteria never had to contend with, an aging star. Measuring the oxygen level of an atmosphere that disappeared 2 billion years ago sounds impossible. You cannot sample air that is gone. You cannot run an instrument through ancient sky. The atmosphere from 2 billion years ago left no direct sample anywhere. Every molecule of it has long since reacted, moved, or changed form, but it left a chemical record in rock, and scientists have learned to read it with extraordinary precision. The most powerful method uses iron chemistry in ancient sedimentary rock. Iron exists in two main chemical states. One state is soluble and moves freely through water. The other state forms when oxygen is present and reacts with iron, creating compounds that are insoluble and sink to the seafloor. By measuring the ratio of these two iron states in ancient rock, scientists can calculate the minimum oxygen concentration required to produce that ratio. The technique is called iron speciation analysis, and it works because the ratio is sensitive to oxygen concentration in a predictable, quantifiable way. Small amounts of oxygen produce characteristic iron ratios. Higher oxygen produces different ratios. The rock records the oxygen level of the water when the sediment settled, Scientists confirm this method against modern environments where the oxygen level is known, proving that the iron ratios in rock accurately reflect the oxygen conditions when the rock formed. Applied to ancient rock, the method produces reliable minimum estimates of ancient oxygen concentrations. Results from this technique filled in the gaps in the oxygen timeline. The oscillations during the 200 million year instability period show up in iron speciation data from multiple continents. The eventual stabilization is recorded in rock layers as the iron ratios shift and hold. The progressive oxygenation of the deep ocean appears as the iron chemistry in deep water sediments changes over hundreds of millions of years. One specific finding from iron speciation stands out. Scientists found that the deep ocean remained oxygen poor for hundreds of millions of years after the atmosphere had become oxygen-rich. The oxygen in the atmosphere did not penetrate to deep ocean water quickly. Surface water oxygenated first. Intermediate depths followed. The deep ocean, below roughly half a mile, held onto ancient anoxic chemistry far longer. This delayed oxygenation of the deep ocean had biological consequences. The organisms in deep water environments had more time to adapt slowly, rather than being hit suddenly by high oxygen concentrations. Some researchers think this gradual oxygenation of the ocean depths may have been a factor in allowing complex life to develop without being overwhelmed by the chemical shock that destroyed the surface anaerobic world. The iron in ancient rock is still being analyzed at sites around the world. Each new study adds resolution to the timeline. The history of Earth's oxygen is not a story told from a few data points. It is a story assembled from thousands of chemical measurements, each one reading a different line from the same 2-billion-year-old archive. And that archive points in a direction that most people have never considered, because the same element at the center of this entire story, the sun, is changing, slowly, permanently. And its change will eventually undo everything the cyanobacteria built. A single question divides the research community on early Earth oxygen. Was the oxygen that appeared 3 billion years ago spread across the whole planet, or was it confined to tiny local pockets? The answer might seem like a technical detail. It is not. The answer determines whether life on other planets would be detectable at 3 billion years into their history, or invisible until they hit their own version of the Great Oxidation Event. It changes how scientists design instruments for studying distant atmospheres. It changes how we interpret every future oxygen signal we detect in space. The evidence from Crow's South African paleosol data shows oxygen at a specific site. The molecular clock data suggests oxygen-producing organisms were active globally in some form. But neither dataset definitively proves global oxygen distribution. They confirm biology was running oxygen chemistry. They do not confirm that chemistry was generating oxygen everywhere on Earth simultaneously. The leading scientific view, classified as a well-supported theory with ongoing debate, holds that early oxygen existed in biological oases, small, isolated regions of shallow water, where conditions aligned perfectly for cyanobacteria, or their predecessors, to operate and accumulate oxygen locally. These oases would have been separated from each other by stretches of ocean where oxygen chemistry was being consumed faster than it was produced. The evidence supporting this view comes primarily from the absence of global oxygen signals at 3 billion years ago. If oxygen had been globally distributed at that time, it should appear in rock formations from multiple continents at the same horizon. The evidence so far is concentrated in specific sites, consistent with localized production. Evidence against this view points to the molecular clock data showing the oxygen-producing community was already diverse and widespread at 3 billion years ago. Widespread organisms typically produce globally distributed signals. The lack of global chemical signal might reflect the efficiency of the planetary oxygen sink rather than the localization of production. Both interpretations are scientifically defensible. The debate is unresolved. Here is why this matters beyond Earth. When astronomers search for signs of life on planets orbiting other stars, oxygen in the atmosphere is one of the primary targets. If early Earth's oxygen was locally confined for 600 million years before becoming detectable at a global scale, then planets with developing life might look dead for billions of years before showing a clear signal. We might already be looking at planets with oxygen producing life and missing the signal entirely because the oxygen has not yet escaped its local oases to become globally detectable. The question of whether early Earth oxygen was local or global is a question about how hard life is defined in the universe. And the answer, when scientists finally confirm it, will reshape the strategy of every space telescope designed to hunt for life beyond our solar system. Earth is the only planet we know for certain has life. That makes it the single most valuable reference point scientists have for designing the search everywhere else. Every method astronomers use to look for life on distant planets, every instrument, every signal, every chemical target, is calibrated against what we know about Earth. And what the oxygen story of early Earth revealed is that the relationship between life and its chemical signatures is far more complicated than anyone assumed before Crow's soil discovery. The standard model for decades was straightforward. Find oxygen in a planet's atmosphere. It is a strong sign of life, because oxygen is reactive and disappears quickly without continuous biological replenishment. No geological process can maintain an oxygen atmosphere indefinitely, so oxygen equals biology. That reasoning is still valid. The core logic holds, but the early Earth evidence adds a crucial complication. Even when life is actively producing oxygen, the planet itself can consume that oxygen faster than it escapes to become globally detectable. Earth had oxygen-producing biology for 600 million years before that biology created a globally observable atmospheric signal. For a distant observer watching Earth from another solar system 3 billion years ago, the planet would have looked dead. No oxygen signal. Nothing unusual about the atmosphere. A world that appeared as sterile as Mars. They would have been wrong. This realization fundamentally changes the odds calculation for how many planets with life might exist without showing detectable signs. If planets routinely go through an oxygen hiding phase, lasting hundreds of millions of years, the number of inhabited planets in the universe that look uninhabited at any given moment could be enormous. Scientists working on the next generation of space telescopes are incorporating this finding directly into mission design. Instruments being planned for the late 2030s and beyond are being calibrated to detect not just oxygen, but the combination of chemical signals that might indicate a planet in its pre-oxidation phase. Methane and oxygen together in certain ratios, for example, is a signal that is hard to produce without biology, even before a full great oxidation event occurs. The South African Paleosol discovery and the molecular clock findings gave astrobiologists a new framework. Life on a planet might be ancient and active long before its atmosphere gives it away. The silence of a planet's atmosphere is not evidence of emptiness. Earth was the proof, and while scientists expand the search outward using Earth as a template, the planet itself is approaching a point in its own story that closes a loop no one expected, because the same star that provides the energy for all of Earth's life is also the force that will eventually end it, and the process it will use is nearly identical to what happened 3 billion years ago, running in reverse. The Sun is not the same star it was when Earth formed. Stars age. They burn through their fuel slowly, and as they do, their internal structure changes in ways that make them emit more energy. The Sun today is about 30% brighter than it was 4.5 billion years ago. That increase is what drove temperatures on early Earth high enough to support liquid water despite the thinner greenhouse effect after the methane disappeared. And the Sun is not done changing. Scientists at Toho University and the Georgia Institute of Technology built climate and atmospheric models projecting Earth's future based on the Sun's expected evolutionary path. The findings are confirmed by multiple independent modeling teams and represent the mainstream scientific view on Earth's long-term fate. In approximately 1 billion years, the Sun's increasing energy output will begin pushing Earth's surface temperatures higher. The initial effect will be on the water cycle. Higher temperatures will accelerate evaporation. More water vapor enters the atmosphere. Water vapor is itself a greenhouse gas, which amplifies the warming. The cycle reinforces itself. As temperatures rise, carbon dioxide in the atmosphere begins to break down. The chemistry involves the weathering of rock, which accelerates in heat and rain. Increased weathering pulls carbon dioxide out of the atmosphere and locks it into rock faster than volcanic activity can replenish it. Carbon dioxide levels in the atmosphere drop. For plants, this is a death sentence. Plants require carbon dioxide to run photosynthesis. Below a certain concentration, photosynthesis stops working. The threshold at which most plants fail is a carbon dioxide level far above zero, but far below what currently fills the atmosphere. As the sun gets stronger and drives more weathering, the carbon dioxide concentration will drop toward and through that threshold. Plants will begin dying. Forests will fail first, then grasslands, then aquatic plants in the ocean shallows. This is confirmed science, not speculation. The physics of solar evolution, the chemistry of rock weathering, and the biological requirements of photosynthesis are all well understood. The timeline of 1 billion years carries some uncertainty, but the direction and the mechanism are solid. Without plants, the primary producers of oxygen on modern Earth vanish. Cyanobacteria in the ocean will persist longer, but they too depend on carbon dioxide and will eventually face the same carbon dioxide starvation as surface temperatures drive faster ocean chemistry. The oxygen supply to Earth's atmosphere will stop being replenished, and oxygen, without replenishment, does not stay in the atmosphere indefinitely. It reacts with rock, with volcanic gases, with organic material in the soil. Without biology actively replacing it, it disappears. The rate at which oxygen disappears from the atmosphere without biological replenishment is actually surprisingly fast in geological terms. The timeline for what happens next, once oxygen production stops, is shorter than most people expect, and the result is something that rewrites the entire future of life on this planet. Plants are not just decoration on Earth's surface. They are the engine that keeps oxygen in the atmosphere. Every leaf on every tree, every blade of grass, every patch of ocean algae is running the same photosynthesis chemistry that cyanobacteria invented 3 billion years ago. The oxygen in the air you breathe right now was released by photosynthesis within the last few thousand years. Oxygen does not last indefinitely in the atmosphere. It gets consumed by reactions with rock and organic material constantly. Biology is perpetually replacing it. Stop the replacement, the oxygen disappears. Scientists modeled the timeline of atmospheric oxygen collapse following the death of plant life driven by the sun's increasing energy. The results are striking. Once photosynthesis stops across enough of the planet to cross a production threshold, oxygen levels begin dropping on a timescale that is fast by geological standards. The model projects that oxygen concentrations could drop to levels incompatible with complex animal life within 10,000 years of photosynthesis failing across most of the planet's surface. 10,000 years sounds like a long time by human standards. In geological time, it is instantaneous. The same system that took 200 million years to build could collapse in a period shorter than recorded human history. The reason the collapse is rapid comes down to the same iron chemistry that played out in the ancient ocean. As oxygen levels drop, chemical reactions that were previously running in the oxygen-rich direction reverse. Iron in rock and soil, previously oxidized, begins reducing again. Reduced iron is hungry for oxygen and reacts aggressively with it, accelerating the removal of whatever oxygen remains in the atmosphere. The process feeds itself in the downward direction exactly the way the ice albedo feedback fed itself in the cooling direction during snowball Earth. The atmospheric oxygen level will plummet. Scientists project the final oxygen concentration during Earth's future deoxygenation at around 1/1,000,000th of current levels. Picture today's oxygen atmosphere as a full tank of gas. The atmosphere of future Earth will have roughly 1 drop left in it. Complex life, which requires the atmosphere to be far above that level to sustain respiration, will be gone long before the concentration reaches that low. The human body starts experiencing oxygen deprivation effects at around 17% oxygen concentration compared to the current 21%. The level at which humans lose consciousness is around 6%. The level at which complex animal life in general becomes impossible is estimated at several percent. Future Earth will drop far below all of these thresholds. The planet that hosted hundreds of millions of years of animal life will return to something ancient, a world of bare rock, chemical atmosphere, and microbes. And the microbes that will inherit that world are the same type that owned it before cyanobacteria changed everything. Earth is heading back to its beginning. The atmosphere that future Earth will carry as oxygen disappears will not be random. The chemistry of a deoxygenated atmosphere is well understood because Earth maintained exactly that chemistry for its first 2 billion years. Scientists can project forward by looking backward. As oxygen leaves the atmosphere, the balance of gases will shift toward what was present before the Great Oxidation Event. Carbon dioxide will remain at low concentrations because the sun's increased energy will be driving heavy, weathering that removes it. Nitrogen will persist as the dominant gas, as it was in the early atmosphere. Methane will return. The methane will come from the anaerobic microbes that retreated to the margins when oxygen arrived. As oxygen concentrations fall and the chemical pressure against anaerobic metabolism eases, these organisms will expand back out of their refuges. Deep sediment communities will spread to shallower environments. Gut microbial communities in any remaining animals will shift in composition. The ancient chemistry will return to the surface. Methane from anaerobic metabolism will accumulate in the atmosphere as it did before cyanobacteria appeared. Combined with the increased solar energy that started the whole process, the methane greenhouse effect will drive temperatures higher. The planet will become hotter, more toxic, and increasingly hostile to any complex life that still clings to existence. Scientists model the future atmosphere of Earth as a high-temperature, methane-rich, low-oxygen environment. The orange sky of the early Earth will return. The oceans, assuming liquid water persists, will lose their blue color as ocean chemistry shifts back toward iron-rich, anoxic conditions in deeper layers. The planet will look more like early Mars than like modern Earth. This future is confirmed science. The atmospheric chemistry, the solar evolution, and the biological responses all follow from well-understood physics and chemistry. The timeline carries uncertainty, but the direction is not in question. Here is the piece of this that is genuinely staggering to sit with. The oxygen window on Earth, the period during which the atmosphere carries enough oxygen to support complex life, began roughly 600 million years ago when animals first appeared. It will end in roughly 1 billion years. That gives complex life a total window of about 1.6 billion years on this planet. The universe is 13.8 billion years old. Earth formed 4.5 billion years ago. Complex life appeared roughly 600 million years ago. Humans appeared about 300,000 years ago. We exist at a point so close to the middle of that 1.6 billion year window that it is almost impossible to visualize the scale. And the conditions that will end the window are already in motion. The sun is already slightly warmer than it was a billion years ago. The chemistry is already running. The clock is counting. What happens to life as that clock runs down is not a single extinction event. It is a sequence, playing out over millions of years, species by species, group by group, until the planet belongs entirely to the same organisms that first learned to survive without oxygen. The future extinction of life on Earth will not happen all at once. It will happen in layers. The most complex organisms die first. The simplest survive longest. Life will be stripped back, category by category, over millions of years, like peeling layers off a structure until only the foundation is left. The first category to go will be large animals. Mammals, birds, and large fish all require high oxygen concentrations to sustain their metabolisms. Their body sizes demand it. As oxygen levels drop from the current 21% toward lower concentrations, large animal species will face increasing physiological stress. High-altitude environments, where oxygen is already thin, will become uninhabitable first. Then temperate zones, then the tropics. The most oxygen-dependent species will disappear before oxygen drops even a few percentage points. Humans, with their large brains and high metabolic demands, are among the most oxygen-dependent organisms on Earth. Scientists project that humans and other large mammals would face extinction well before oxygen concentrations drop to the levels that eliminate most other animal life. After large animals, smaller insects and invertebrates will persist longer. Their oxygen requirements are lower per unit of body mass. Their simpler metabolisms tolerate a wider range of atmospheric conditions. Some insects can survive at oxygen concentrations that would render a mammal unconscious. They will inherit the warming, oxygen-depleted world for a period after complex vertebrates are gone. Eventually, even they will face concentrations too low to sustain their chemistry. Fish in the deeper, cooler parts of the ocean will persist longer than land animals, because cooler water holds more dissolved oxygen than warm water, and the ocean temperature in deep zones will rise more slowly than surface temperatures. But as the ocean heats and oxygen in the surface layers drops, the deep ocean will eventually follow. After animals, plants, and complex algae will already be gone, having started the collapse by dying from carbon dioxide starvation. Fungi, which decompose organic material rather than producing their own food, will hang on longer than plants. They do not require photosynthesis and can tolerate lower oxygen levels. The last complex, multicellular life to survive will likely be certain types of simple worms and small invertebrates in deep ocean sediment, where conditions change most slowly. Scientists confirm these types of organisms have extremely low oxygen requirements and can survive in nearly anoxic conditions for extended periods. Below them in the sediment, the anaerobic bacteria will already be thriving again. The sequence from the first animal extinctions to the last complex organisms disappearing will span hundreds of millions of years, plenty of time for each category of life to reach its limit and fade. The planet will grow quieter, simpler, and older looking with each passing million years, until only the microbes are left. And the microbes that will dominate the final Earth will belong to the same lineages that have been waiting, quietly, in the dark sediment and deep rock since the great oxidation event pushed them there. They outlasted everything before. They will outlast everything after. Earth's story runs in a circle. The first life forms on this planet were anaerobic microbes, simple single-celled organisms that ran their chemistry on sulfur and iron in a hot, oxygen-free world. For 2 billion years, they were the only life Earth had. Then cyanobacteria changed the atmosphere, and everything that came after, every plant, every animal, every human civilization, was built on the oxygen those microbes produced. Now that oxygen window is closing. The sun is getting stronger. The plants will die. The oxygen will disappear. And when it does, the anaerobic microbes that retreated to the margins will expand back into the world they were forced to leave. The last living things on Earth will be functionally identical to the first. Scientists confirm this through both biological projection and the rock record. The anaerobic bacteria in deep sediment and volcanic environments today carry essentially the same core chemistry as the earliest microbial life on Earth. They have diversified in detail over billions of years, but the fundamental strategy, running metabolism without oxygen, remains unchanged. These organisms survived the Great Oxidation Event, survived every mass extinction since, and will survive the final deoxygenation. They are the most durable life forms Earth has ever produced. When the oxygen is gone and the atmosphere returns to a methane and nitrogen mix, these organisms will not be struggling to adapt. They will be returning to the conditions their chemistry was originally built for. The restoration of anoxic conditions is not a crisis for them. It is a homecoming. Picture the full timeline, 4.5 billion years of Earth's history. The first 2 billion years belong to anaerobic microbes alone. The next 2 billion years belong to a world slowly transformed by oxygen, building toward complexity. The final 1 billion years sees that complexity disassembled, oxygen returned to near zero, and anaerobic chemistry reclaiming every available environment. The organisms at the end of Earth's story will breathe the same atmospheric chemistry as the organisms at its beginning. They will run the same basic metabolism. They will live in the same types of environments. A microbiologist from the distant future, if such a thing could exist, would look at the life on the final Earth and see something nearly indistinguishable from the life on the earliest Earth. 3.5 billion years of biological history, and the planet comes back to where it started. The window in which this planet was capable of supporting anything more complex than a single-celled organism is a brief feature of Earth's story, not its defining character. The defining character is microbial. It always was. And the thin, unlikely, improbable window of complexity that produced everything from the first fish to human beings, all of it resting on a foundation of ancient bacterial pollution from organisms that had no idea what they were doing, turns out to be one of the most extraordinary coincidences in the 4.5 billion year history of this planet. You are breathing the most unlikely air in Earth's history. The oxygen in your lungs right now exists because of a chain of events so specific, so dependent on exact timing and exact chemistry, that if almost any single piece had gone differently, you would not be here. The planet you live on spent 2 billion years with no breathable air. It will spend its final billion years with no breathable air. The window in between is where we exist. That window is approximately 1.6 billion years wide, if you count from the first complex animals to the projected end of oxygen. Humans appeared 300,000 years ago. We are not at the beginning, the middle, or the end of this window in any rough sense. We are somewhere inside it, closer to the middle than to either edge, which sounds reassuring until you remember that the forces closing the window are already running. The sun is already warmer than it was a billion years ago. The rate of solar brightening is slow by human standards, but relentless by any standard. The weathering that will eventually starve plants of carbon dioxide is already happening. The chemistry is not waiting for a start signal. It started 4.5 billion years ago and has been running continuously since. Scientists confirmed the 1 billion year estimate for when oxygen production will fail through multiple independent climate models. The exact number carries uncertainty in the range of hundreds of millions of years in either direction, but the mechanism is solid. The sun will get stronger, the carbon dioxide will drop, the plants will die, the oxygen will follow. On that scale, 1 billion years feels like a long time. By any human measure, it is incomprehensibly long. But the story this video told should put that number in perspective. 3 billion years ago, the first oxygen-producing organisms appeared. It took 600 million years for their oxygen to overcome the planet's natural sinks. Then the atmosphere fluctuated for 200 million more years before stabilizing. Then, hundreds of millions of years passed before complex life appeared. Then another few 100 million years before animals like us existed. Every step took longer than anything in human experience. Every step nearly did not happen. The molecular clock could have stopped at a different branch. The South African oases could have been consumed without leaving a trace. The 200 million year oscillation could have locked in the wrong direction. The 1 billion year oxygen window could have been 0 years long. It was not 0, it was long enough. And inside it, against enormous odds stacked by chemistry and geology and time, the universe produced creatures that can look back at the ancient rock, read the chemical diaries sealed in salt and iron, trace the molecular history of every protein in their own cells, and understand, with something close to certainty, how all of it happened. The orange sky is gone. For now, the green ocean is gone. For now, the planet that would kill you in a breath is gone. For now, but it remembers what it was, and it is, slowly, becoming that again. The window is open, we are inside it. And the light coming through it is the most improbable, most fragile, most extraordinary thing in 4.5 billion years of this planet's existence.